I'm going to take the calm for a little bit and give you this initial glimpse that we promised you of the Knowledge Center, what we're trying to do to translate a lot of what you heard today through the DCC for the Metabolomics Workbench and from Christy and George, and build a resource to some sort of being a jumping off point for the entire ecosystem. That's really what we're hoping to do. And I'm going to kind of cut my time a little short because I really want you to hear from Kyle Gaulton, who's one of the other MPIs on the Knowledge Center, but also is, to me, one of the ideal users for the Knowledge Center, because he is an investigator all by himself who thinks about, scientific questions related to metabolism and diabetes. And he works in the functional genomics space. And he is, to me, a perfect user because he may not have known what all these resources were. But as we started working on this project, it was really cool because as he sat in the room, he did exactly what I'm hoping that we can do with you all on these calls, is go, ooh, what if we did this analysis? He's going to tell you a little bit about that in the end. So to begin, the Knowledge Center. Our initial plan was to bring scientifically valid results to the wider research community. We had 5 aims to do that. Well, to build a first blush of that, it's a lot to tackle, right? There's a lot of resources. Even just listening to Mano and Shankar talk about the richness of Metabolomics Workbench, it's a lot. They have an entire community, entire set of investigators, data, approaches that they house and really are the shepherds of. How do we as a Knowledge Center shepherd that to a broader community? It's a bit of a challenge, but here's how we decided to approach it. So I don't know if you know, many of us that are on the Knowledge Center build scientific resources also for human genetics. And this is an important example, I thought, because it kind of sets the tone for how we approach the Knowledge Center. For example, if you are a human geneticist and you have a favorite phenotype and you want to generate a list of genes to investigate, in a world where you don't have all these resources, you might go to one resource, the GWAS catalog, download all the summary statistics, run a meta-analysis in R, knowing all the proper scripts and processes and tools and steps you want to do. And then you're going to incorporate a bunch of other resources. You're going to go to this thing called FUMA GWAS. You're going to do literature searches. You're going to go to model organism databases. You're going to use different resources that provide gene expression results. And then finally, at the end, you may have a list of genes to investigate. You'll do this well. You know what you're doing. You're an expert. But sometimes you'd like it done for you and a little bit easier so that if you want it represented in one place, very simple, you can have it access to you. That's what we build with our human genetics resources. We don't take away all those details in between. In fact, we hope that you would want to go back and do those to sort of verify, but we make it accessible. And really what we try and do is we catalyze research using the 80-20 rule. But for those of you who like food and live in New England, here's the example I like to give. It's called the lazy man's lobster. If you love lobster and you normally eat lobster, you realize it's a messy task. It requires the bib and tools and butter, and it's messy, and there's a lot of things to do, and then you finally get to eat. But they have this thing called the lazy man's lobster, where the beautiful, elegant cooks behind you, they prepare this beautiful plate of butter lobster served directly for you, which sometimes suffices, even if you're an expert and you know exactly how to deal with the lobster. So I want you to keep that in mind when you think about how we built the Knowledge Center. We want to make so they can readily accessible to you, even if you're an expert in that data type or in that scientific question. But if you want to dig deeper, you can, but you can get the results right to you through the Knowledge Center. So how do we approach this? Well, we started with listening, doing research, and really collaborating with all of these data ecosystem members in the DCCs over the last 6 months. And we realized, wow, there are a rich host of data resources and tools. It's incredible. We have a lot of learning to do. And you as a user must have a lot if you actually wanted to take on the task of learning these. So how can we make this a little more simple for you? Well, what we learned is, even in the ecosystem, as investigators inside the ecosystem, we all wanted to put the information about all of these DCCs, what they do, in one place, so you can find them. We want to make those results and data more meaningful to an end user. We also want to enable things like more of the data being used, because some of the resources said only 1% of our data is used by 90% of our resources. We want to contextualize all of these DCCs in human disease context or in basic biology. We want to make sure they're sustainable, and we really want to use the resource to say what can be done with all of these data resource centers. So we attempted in the last 5 months, we're very new to this effort, we're very excited to be part of it. We attempted to get water flowing through the system. I know that sounds silly, but that was really the goal. And we really want to make it accessible. We want to be vulnerable. In many cases, a lot of times people go off and they build resources and they build this beautiful, elegant system, and they're not sort of dipping their toes in the water to the researchers. And then it may not be exactly what you need. And if it's not exactly what you need, you may never come back. So our approach is really, we want to do this frequently and often and share with you what we've built and see if it's right. Tweak it, erase the whiteboard, start over. So what you're going to see today is our first pass at presenting the ecosystem in an interactive way, allowing you to navigate through the DCCs and represent results from a very few scientific vignettes that we've come up with together in a context of, say, a drug developer, an experimental biologist, or just the average person who's curious. So with this initial offering, we have all the information of all the DCCs curated in one place. We make it easy to get to. We provide like a jumping off point, a start, like on the cliff, right? And we hope this resource will reach a broader research context. So how do we do this? Well, we want to organize knowledge in 4 different categories. Without going into these in the interest of time, I'm going to basically more explicitly describe them. One is we want to present results if they've been generated as is by a specific DCC. This is they've done the analysis, they've done the results. It's very simple. We want to make that accessible rather than having you go to that specific DCC. We also, in the case of the Metabolomics Workbench, as you heard today, a lot of the stuff you can do in Metabolomics Workbench, but you need to know what you're doing, right? You need to be an expert in metabolomics. What if we could pre-compute some of those for you in the context of human disease or your favorite metabolite? That's another vignette we'll tell you. What if we could generate results that the DCC itself would love to be doing, but may have published in a separate publication, has the data sitting on their DCC. All we need to do is run that method and present it to you. That's an example of GTEx you're going to hear about. And then finally, the most complicated one, which gets to, I think, Hunter's point a little earlier, which was perform a new analysis that's sparked by a scientific question. So take data from a resource, run an external method that creates new results from that, then add an external resource like genetic associations from the largest repository for genetic associations out there and present results in the context of biology for the first time. So that's what you're going to hear about. That's what we've built. We're just beginning and it's not perfect, but we want your feedback. So that's the whole thing is contextualized by human questions. And so that's what we're hoping is if you have questions, if like from this webinar, one person is like, I would love it if they could insert blank. Run this analysis and produce these results from this DCC or this set of DCC resources. We'd love that. And that's what we want to do more of. The team that built all this is my fellow MPIs and this wonderful team. We built it in 6 months, which is pretty awesome. And it's just the beginning. So with that, I'm going to give you a little tour and then I'm going to hand it over to Kyle to give a more specific tour. If you want to log in, it's cfdeknowledge.org. Remember, it's new. We want feedback, but we want you to take a look at it. It's meant to get us all organized, give us a gateway to spark your curiosity and contextualize your research. I'm going to look at the time, make sure, oh, I have 6 minutes. Holy moly. So I'm going to first start with just giving you a view of the landscape of the page. And then I'm going to quickly turn it over to Kyle to make sure that we get some time. So here's the page if you land on it. The idea is to give you sort of a glimpse of the ecosystem from the perspective of areas of biology or research. So the idea is if this page is interactive, it's meant to be sort of a navigational tool, but also sort of an exploratory thing to learn about the components of the ecosystem. So if you click on each of these, you'll see these dotted lines that connect to the DCCs. And they're thick and thin based on how relevant they are to that sort of context of study or research. This is not a perfect science, but this is our first attempt to sort of put this into a framework for you all who may not be familiar with what MotorPack is, for example. So if you hover over them, you'll see them change and whatnot. Then you can set your context and your entire experience can come after that. What we do then is if you set your context and you hover over each of these DCCs, you get a description of what these DCCs do in the context of that particular research frame. What's also nice is if you'll see the orange highlighting below, you'll see for that DCC, the omics data sets they have, and then how they organize their data, whether it be by different entities, whether it be by gene, variant, protein. And this is sort of how we imagine you as a researcher navigating through the rest of the resource, right? Because these DCCs span things that have in common, but they also do it in their own way. So you can choose to navigate the resource that way, but you can also choose to learn this as a learning opportunity. I'm going to turn it over to Kyle real quickly and not do 2 gene searches because I think Kyle will be best to do that. But if you want to learn anything about everything we've done so far, you have access to all of our webinars, description of the research context that we've done, so how we define those, documentation, how we've done each of these analyses, and then information on each DCC. This is what we meant by everything in one place. This is a lot, but it's also a start. It's the idea that each DCC is contextualized by the same things I told you on the front, and you can navigate and pull and play based on those. But you can learn about the foundational, primary, and secondary, and even tertiary knowledge generated in those resources but also what's available in the KC. So with that, I'm going to turn it over to you, Kyle, to give a couple stories, particularly around Metabolomics Workbench and 4DN, that will be most relevant to this audience in the last couple minutes. That's okay.
Sure. Okay, I'll try to fit that into 2 minutes.
Sorry.
Okay, so I'm actually just going to talk about the Metabolomics Workbench results very quickly. So based on what Shankar and his group talked about, they have collected metabolomics data and specifically provide the results of studies that have compared metabolite profiles in various conditions. And one thing that we thought would be valuable to collect for the Knowledge Center was a meta-analysis of associations of metabolite levels for the same trait or disease across this resource and provide that on the trait page on the Knowledge Center. So briefly, what questions can we ask from metabolite data of case control samples? So this may differ depending on the type of user you are, but if you're, in pharma, drug development, for example, what metabolites might represent biomarkers of a disease? In other contexts, can these metabolites give us insights into the underlying mechanisms of disease? If you, for example, go to the type 2 diabetes page, on the Knowledge Center and look up metabolite associations meta-analyzed across the data in the workbench. This will show you a plot of the associations ranked by p-value. And if we look at the top associations for type 2 diabetes, there are some that make sense, like glucose, for example, as some of the top results for type 2 diabetes. And the URL is here, but you can also go search on the Knowledge Center for type 2 diabetes, and this is one of the modules that you can see results for. So can this give us novel insight into metabolites that are associated with disease? So in several cases, I just pulled out a couple of examples of where these associations may provide useful information for understanding metabolite levels in disease. So for Alzheimer's disease, for example, glycine was one of the top associations with Alzheimer's disease compared to controls. And glycine has had some evidence, although not definitively, for a potential role in Alzheimer's disease. Another example that I thought was interesting, Amaralab studies type 1 diabetes is one of our primary focus. And in the case of type 1 diabetes, if we look at metabolites associated with type 1 diabetes, there are several top molecules. I'm not going to try to pronounce this, but the abbreviation is LPC. It's a lipid, which has altered levels in T1D, as well as, in this case, prior to the development of T1D. So maybe a biomarker of future development of T1D. It's also implicated in many processes that drive type 1 diabetes risk in a variety of ways, and therefore maybe a metabolite that is interesting to pursue further for involvement in type 1 diabetes. So those are just a couple examples. There's other traits that have similar association results that you can look up and gives you insight into metabolites that's summarizing data across the metabolite workbench. So we are interested in further improving these associations in a variety of ways. I'm not going to belabor this too much, but just to say that this is the first example of combining data from Workbench, and we're continuing to work with them to further enhance these results. For example, to change the types of analyses we do so that it provides effect size and direction, as opposed to just p-values, as well as maybe thinking about untargeted metabolite data, where the metabolite is a bit less clear, but it's a larger set of metabolites you could profile. And so thinking about how we can move beyond just targeted data, which is what we've used to date, where you know the metabolite, to thinking about how we can incorporate untargeted data sets in these analyses as well. So I'm just going to end then on thinking about how we can build on what we've done here beyond just the question of, are these metabolites associated with disease and levels of these metabolites. So one question we're quite interested in, given our expertise in genetics, is what disease-associated metabolites are also involved in causally in the risk of disease. And there are a series of resources we could consider using, some internal to Common Fund, some external, to address these questions. For example, QTLs for metabolites, so variants that affect metabolite levels. Are those variants also associated with disease risk? 2, what's the tissue cell type activity of these metabolites using expression profiling, also inferring metabolite sensor pairs, which are analyses you can do from single cell data, for example, to understand the potential role in these tissues of metabolites that are disease associated. And then 3, what metabolites represent early biomarkers of a disease and phenotype, and this could utilize data at pre-disease states, some of which is in Metabolite Workbench, but may require slightly different analyses. Other resources in Common Fund, for example, A2CPS and others, are collecting metabolomics data in preceding stages of disease that could be used to address whether altered level of a metabolite is a biomarker of future development of a disease. So those are just some questions. I'm sure there are others, and yeah, it would be great, to get feedback from other types of questions that people would want to ask in this space, and combining data from either Common Fund resources and other external resources as well. But these are just some of our thoughts on what we could go from here. Okay, so that was fast. I'm sorry, but I wanted to make sure I stuck to time. And yeah, thanks very much. And yeah, like I said, we're continuing to think about how we could enhance the representation of these data and other data sets on the Knowledge Center, as well as what are the other types of analyses that could draw in multiple different data types that could be useful to users of the resource.
Beautiful. Thank you, Kyle. Sorry we didn't give you enough time.
No worries.
So with that, actually, I want to quickly share my screen and just invite you. Thank you for hanging in there a few more minutes longer, but we're just at the beginning. So next month, you're going to hear from our Data Resource Center. In addition to Avi, you're going to hear, Shankar, who you heard from today, you're going to hear from Avi Mayan directly and talk about the Data Resource Center. And I think this is really cool because it's a lovely complement to what we're trying to do in the Knowledge Center. And this is sort of an image that I generated when I was thinking about the DRC. It's sort of analogy to what I show with the child sort of walking through the ecosystem. I think the DRC has this sort of complex, a playbook. And the idea is that you can see all these different components of the Common Fund data ecosystem, and it provides you with all the data, all the assets in one place if you want to generate your own analysis. So different than the Knowledge Center, when we expose it to you already pre-cooked, a la the lazy man's lobster, with the DRC, you can actually dive in and do it yourself. And so you'll hear about that next month. And this is just a sneak peek of this really cool resource that Avi and his team have built that allows you to build your own sort of way of walking through the data ecosystem and build your own sort of playbook of questions you might like to, scientific resources you'd like to access in the context of scientific questions. So we'll hear more about this in a month. And if you want to join our mailing list, please email this listserv, help@cfdknowledge.org. And it comes to like all of us. So that's great. So we respond really fast. The recording will be found from today on our YouTube channel, and I'll send the link around, but that's it. And also, if you just want to email me, Burtt, B-U-R-T-T, @broadinstitute.org, you can always ask us any questions. If you have any ideas for analyses, we really love to hear from you. And we're hoping that you tune in for the future webinar, which will happen in a month's time. Mackenzie, what date is it? It's escaping me right now. May 9th?
If you go back to Avi's slide,
oh, it's there.
It has it there, but it's, I believe, May 30th.
May
31st, sorry.
So it's after Memorial Day weekend, so hopefully everyone will be back. So I want to thank our speakers. This was really great. We packed in a lot to you today, but I appreciate you all hanging in there, and I appreciate all the speakers' time and careful sharing of really cool data. Thanks, everyone. Take care.
Thank you.
Thank you.
Bye.